So today we are going to discuss about Module 4 Biological Sequence Analysis, so which consists of DNA sequence models, independent identically distribution model, Markov chain model, matrix association regions, subsequence pattern models, regular expressions, weight matrices, blossom and position specific scoring matrix. So first we have to see the introduction of biological sequence analysis. Biological sequence analysis involves studying the sequence of biological macromolecules like DNA, RNA, and proteins. The primary goal is to interpret these sequences to understand their structure, function, and evolution. Types of biological sequences: DNA sequences composed of 4 nucleotides A, T, C, G. So DNA encodes the genetic information. RNA sequence similar to DNA but with uracil instead of thymine. RNA play roles in transcription translations. Then protein sequences made of amino acids proteins perform the numerous functions in cells from catalyst to structural roles. So for example, DNA alphabet is a summation DNA which consists of A, C, T, G. So if you take the add all DNA sequences is equal to 4. The next we are moving on to the sequence models. In biological sequence analysis, understanding sequence models helps researchers make prediction about the nucleotide or amino acid sequences. Two important probabilistic models used to describe sequences are the independent and identically distributed model and Markov chain model. The two main sequence models are independent identically distribution IID model and Markov chain model. Independent and identically distributed model assumes that each position in the sequence is independent of all others and that every nucleotide or amino acids comes from the same probability distribution. This means the occurrence of one symbol in a sequence does not influence the occurrence of another, and the probability of each symbol remains the same across the sequence. Independence: each element in the sequence occurs independently of the others. So identically distributed means each element follows the same probability distribution. That is, the probability of seeing a particular symbol remains constant throughout the sequence. So when you see the mathematical representation, we have the symbols in the sequence x1, x2 up to xn. The probability of entire sequence is simply product of the individual probabilities. The probability of x1, x2, xn is equal to p of x1 into p of x2 into p of x3 etc. up to p of xn. Here the p of xi is the probability of observing symbol xi, and this probability is same for all I. Example, you consider the DNA sequences. Assume the probability of observing nucleotide A, T, C, G are all equally probability, which means there are 4 sequences, 4 nucleotide sequence, P, probability of A, probability of T, probability of C, probability of G. So we have the 4 sequence. If you take the probability of A, it is 1 by 4, probability of T equal to 1 by 4, probability of C equal to 1 by 4, probability of G equal to 1 by 4. According to the IAD model, the probability of observing specific 4 nucleotide sequence such as A, T, C, G is, then we can multiply all these sequences probability of A, T, C, G, so which gives the 0.00390625. So when you take the percentage, so which means 0.03 percentage. Next move on to the Markov chain model. So a Markov chain is a probabilistic model where the occurrence of symbol at a given position in the sequence depends on the symbol at the previous position. Dependence means the probability of symbol depends on the preceding symbols. Order: Markov chain can be different orders. First order Markov chain and higher order Markov chains. So when you do the Markov chain model, we need to know the condition probability. So the formula for condition probability is probability of A given that B equal to probability of A intersection B divided by probability of B. So when you calculate the probability of A given that B is condition probability of A given B. Probability of A intersection B is probability that both A and B occur. Probability of B is the probability of B occurring. So here we have to know, we understand that probability of A given B is probability of A given B. The B is already happened, so we have to know that we have to find the probability of A. So that is the meaning of probability of A given B. So here for example, if you take the up to x1, x2, xn sequences, the probability of sequence is a first order Markov chain is given by P of x1 comma P of x2, probability of P of x1 comma probability of x2 up to xn is equal to probability of x1 into probability of x2 given x1. Here when you see the second sequence, probability of x1 has already happened. Then you have to write the probability of x2 given that x1 into probability of x3 given to the x3. Then probability of xn given that xn minus 1. So similar way you have to find the when you find the probability of x2 given that x1 or probability of x3 x2, so we know that the transition probability of the every sequences.